So thank you, and it's a real pleasure to be here this morning. I think it's a real celebration of the silver jubilee of Cytel. Before I get on with my talk, I'd just like to say a few things in that regard. I first met Cyrus when I was a graduate student, and I worked in the Dana-Farber Institute for 3 summers, around about 1980. And looking back, that must have been the time that Cyrus was working with Nitin, developing the network algorithms that lay behind StatExact. And that was then the launch of the company. We went separate ways. I came back to Europe. I was working in England, still collaborating with Bruce Turnbull in the States, working on group sequential methods. And in 2000, Bruce and I got together with Cyrus because we'd finished our book, and I think East 3 was coming out then, and we found that we dovetailed very well. East 2000 was it? Okay, for the millennium, and we gave some short courses together that year. And what Bruce and I had been really trying to do was put together material from a big literature to synthesize that into a common framework, common notation, and make the methods accessible. And of course, in East, exactly the same had been going on. There was an awful lot of work out there, but if you wanted to do something, if you read one paper, it was in terms of Z or Z in America, or another paper, it was S or W, and one was for binary data, one was for normal data. But actually everything really follows the same pattern. And a lot of work, I think, was needed to translate that research into something that was readily usable. And it's great to think over the years how much must have happened, how many trials used sequential methods because of the contribution at that stage of bringing software together in a way that people actually could use. And the support that East, for East that Cytel gives, I think, is another big factor. Things don't just come off the shelf and land in people's laps. So I think it's been a marvelous contribution to medical science, to medical treatment, to patients, as well as the whole academic side. And I'm delighted to be in some way a part of that. So let me move on to my talk. How do I get that? Oh, I see. Good. I'm going to talk on a particular topic in adaptive trial design with a survival endpoint. And I hope in the course of this talk to outline the ingredients that you put together to do adaptive trials when there are multiple hypotheses. So there'll be closed testing procedure, there'll be combination tests, but there'll be special methods for survival data. And there's a bit of a twist that comes up in this application. I'll try and go slowly. I know it's early in the morning, and I think if I go slowly enough that I can work out what I'm trying to say, then I hope you can understand what I'm saying. This was based on work I did with Martin Jenkins and Andy Stone at AstraZeneca. We actually worked in a slightly different application, but the key thing about the way we've done combination tests is what's in this talk. So to outline the talk, I'll describe a clinical trial with a survival endpoint, and the point is there's a treatment selection stage. So there'll be 2 experimental treatments, or 2 versions of the experimental treatment to be compared with a control, and at an interim point in the trial, one of those will be chosen as the one to proceed with. And so in order to protect type 1 error, then we have to introduce some special methods, and I'll explain how that's done in terms of a closed testing procedure for dealing with the 2 different hypotheses about either one experimental treatment or the other being compared to the control, and a combination test for combining data across stages. I'll explain a little bit about Logrank statistics and how they proceed when you look at accumulating data. And then putting the ingredients together, and the interesting thing is if you do it in a slightly naive way, it doesn't work. So I shall explain why that happens. And then if you do it in a different way, it does work. And that was the method 2. So, okay, but it's still have to go over, press page down to, which isn't, yeah. Okay, so here's our setting. We have let's say a cancer treatment, which can be given in 2 forms. The first version, let's say in the cycles of chemotherapy, there's a short and intensive treatment period in each cycle. And in the other version, the treatment, the administration, is less intense and takes place over a longer period. And the investigators might think, well, one could be more effective, one might be a safer way of doing things. But actually, it's only going to be when data start being observed on the subjects that one can tell which method is either more effective or more safe. And so during the study, at the interim stage, the data so far will be examined, and one of these treatments will be chosen as the one to continue with. And the endpoint, let's say the primary endpoint, is overall survival. Of course, one of the problems with overall survival is it takes time to get information. You have to see deaths to get information on treatment comparisons. Progression-free survival, progression can happen sooner. You'll have more progression events at an interim analysis. So it's tempting to use progression information to make the interim decision because there's more of it, hoping that progression is a good surrogate for overall survival. And safety information as well will be used at the interim analysis. And then after the interim analysis, only one of these treatments will be taken further, in the sense that recruitment will then be to the selected experimental treatment and control. There's still follow-up on the patients who are already in the study. So here's a trial plan. And the way I've explained it is to say, before the interim analysis, we can talk about the patients admitted thus far as being the stage 1 cohort. So there are patients admitted initially, they're randomized to one of these 2 experimental treatments or the control, and by the time of the interim analysis, there'll be follow-up. There'll be progression-free survival data, there'll be some overall survival data, there'll be safety information as well. The decision is made at the interim analysis as to whether we continue recruiting to experimental treatment 1 and control or experimental treatment 2 and control. And then in the remainder of the study, we get the new cohort coming in there with their follow-up of survival. And we also get continuing information, just following this first group of patients, learning more about their overall survival as they carry on living longer. And at the end, we want to test a null hypothesis that overall survival on the selected treatment is no better than control. And of course, the investigators will hope to reject that and be able to conclude that their new treatment is doing better than control. But in that test, allowance must be made for the fact that this treatment was selected because it was doing well at this early stage. So there's a multiplicity problem. So, bit of notation, if we're protecting the type 1 error rate, well, what are the hypotheses that are being considered? If we assume a proportional hazard model, let's keep things nice and simple, so lambda 1 and lambda 2 are hazard ratios for these 2 comparisons, control versus experimental treatment 1 or control versus experimental treatment 2, and let's work in terms of theta 1 and theta 2, which are the logs, so that we can have a null hypothesis that theta 1 is less than or equal to 0 that would say experimental treatment 1 is not better than the control, and we have the alternative that it is theta 1 positive. And then there's a similar null hypothesis for theta 2, comparison of experimental treatment 2, and the control. And to protect the family-wise error rate, we want to control the probability of rejecting any true null hypothesis. So either of these or both of these might be true, and so if one of these is true and we end up rejecting it, then that's a type 1 error. So that family-wise probability must be controlled at level alpha. So a closed testing procedure gives a very nice way to approach that sort of multiple testing problem. And what you do is, for your two, in this case, there are two null hypotheses. Each of those, we make a level alpha test just by itself. A level alpha test of H01, a level alpha test of H02. And then we also have a level alpha test of the intersection hypothesis that says both H01 and H02 are true. So if they're both true, then this test must have probability at most alpha of rejecting that intersection. Now, these are just the building blocks. What really happens then at the end is you say, can I reject H01 overall? And the answer is yes, if you manage to reject this level alpha test of H01, and if you also rejected this one, this intersection test. And the same thing for H02. Overall, we can reject it if H02 and H012 are both rejected. And the paper is Marcus et al. 1976 that introduced closed testing procedures just gives a very nice little proof that says, well, this is all you need. This makes sure that family-wise error rates are protected. So that's the first ingredient. And if you like, it deals with a selection bias. The fact that in the end, if we've selected treatment 2 to compare, we'll have chosen this because it was doing well at the interim stage. And it's really this intersection hypothesis that's catching that and making sure that we don't inflate type 1 error by a selection bias. The other part of the process we need is a combination test to combine data, say, from before and after the interim analysis. So we've got these three different hypothesis tests going on now, H01, H02, H012. For each of them, we can say we get a contribution before, we get a contribution after the interim analysis. And how do we put those together? Well, a combination test is a very simple way of doing that. We say from the first part of the data, we'll have a Z1, which is standard normal, or I guess if you're further into the null hypothesis, it's smaller still. But if the theta that we're testing is exactly 0, this will be normal 0, 1. And then we need to get the same thing from stage 2. And so the important thing is that even though we've made an adaptation, conditionally on the way that stage 2 has been set up, you sort of start with a clean sheet and say this Z2 will also be a standard normal, or again, smaller than that if the theta is really negative. And if these are both standard normals, we can combine them with some weights, where the W1 and W2 have sum of squares equal to 1, and we get an overall Zed statistic. So we can simply reject the hypothesis, null hypothesis here, if this combined Z is bigger than the upper alpha point of the normal distribution. You can define into the same sort of test in terms of p-values instead of Z values. They obviously just relate to each other in a simple way. So now we're able to, we hope, test our three null hypotheses across the two stages. But we are doing this with survival data. So again, there's an extra bit of thought needed to make sure that we're doing that well. So my little diagram here is meant to illustrate all that goes on in monitoring survival data over time. The patients obviously arrive at different points. There's a staggered entry into the study. So all these timelines for the blue ones are on the control treatment, the red ones, in this case I'm just taking experimental treatment 1 to illustrate. They're all starting at different times. Some of them, this is for overall survival, some of them die, and that's the solid dots at the end. Some of them may be censored, right? Censored. At the final analysis, there'll be just end of study censoring. There may be some loss to follow-up. But at an interim analysis, you just get a fraction of the data, right? Some of the subjects haven't even arrived yet. We don't know they're going to be in the study. Others, they have arrived, but we've only followed them this far. So in this particular case, there's a death up there, but all the next 4 will be right censored at the interim analysis. Some of them will later either fail or be censored at a later point. And what you can do is at the interim analysis, you can just put the data together, sort of set the clock at 0 for everyone to rearrange these timelines so you can think about them for a log-rank test. And then the question is, so how is the log-rank statistic distributed? And when you look at it twice, with the data growing, how do the 2 statistics go together? And the answer is rather neatly. It's easiest to think about this on the unstandardized scale. So this is the log-rank statistic, basically number of failures on one treatment minus the sort of expected number of failures on that treatment, before we've divided by something to make it standard normal. And the information, which is to a close approximation the number of failures over 4, comes into the formula. And what you have is at the first analysis, this log-rank statistic will be normal with mean information times log hazard ratio and variance information. And then if you say, well, what happens at the next analysis, S2, when we've got the extra data? Well, the nice thing is that the change, the increment, S2 minus S1, is normal with the same sort of formula, but we've got the increment, the I2 minus I1 increment in the information. And these 2 things are independent. So it's exactly the same scenario as if we'd had, if I1 was an integer, this many subjects providing a normal theta 1 observation in the first analysis, and then an additional I2 minus I1 new independent ones in the next analysis. So this independent increments property goes back a long way. Cytel showed that in 1981. And it's been proved in sort of more and more realistic situations but it was done by then to have staggered entry and all the possible censoring and so on. So this really means that there's no problem dealing with accruing survival data. It just fits into the standard sort of model. So we can put it into a combination test. So we can say from the first analysis, if we want a Z statistic now, we just standardize our S1, divide by root I1. And then the Z2, which is meant to be from the new information that comes in after the first analysis, we take the increment S2 minus S1, standardize that. That will be our second Z statistic. And we can put them together to do a test. All very simple. There is one condition that has to be met, though, and this can actually be a problem. We need that this Z2 has the right standard normal distribution, regardless of what went on at the interim analysis. And it isn't really the case. I was trying to draw the comparison between a first stage of a trial where you have one set of patients, you get their response, and a second stage where you get a new set and you get their responses. What we're getting here is more information on the same people, as well as some new ones. And because we looked at things at the interim analysis, say progression-free survival, which is a strong indicator of what will happen in overall survival, the people that we're following up, we sort of know a lot about. And the decision that we're following this group rather than that group was based on progression-free survival. We might start tweaking our second part of the trial in a way that could bias things. Martin Posch and Peter Bauer wrote about this in a letter, I think it was, to Stats in Medicine, showing the pitfalls. So here's a scenario that one can imagine. Suppose at the interim analysis, progression-free survival is is rather good for patients on experimental treatment arm number 1. So we think they've got good prospects for overall survival. So those are the patients already in the study. So we're convinced they're going to do rather well and give us good data supporting the experimental treatment. Let's try and take advantage of it. Let's keep them in the study, and let's not recruit too many new ones, because they might water down that good data. Well, you can see that could rather swing things in favor of rejecting the null hypothesis in a way that shouldn't be going on. And it's very hard to say that things like this won't happen if you say, well, we look at the data and we make a decision and we rather adaptively and flexibly decide how to continue the trial. The regulators will say, well, I'm not convinced that you didn't do more than that. So this is a problem. And we've got to see, is it going to crop up in our example when we put all the ingredients together? So let's go back to our trial with the 2 treatments. We're testing these null hypotheses and the intersection hypothesis. We've got combination tests to do it. So one way to explain the whole process is to say, well, in terms of Zed statistics, what are they all? We've got, say, for H01, what's the Zed statistic that we get from the stage 1 data? And then in the other half of the combination test, what's the Zed statistic for that hypothesis that we get from the stage 2 data? And likewise for the other hypotheses. If we just go through and specify them all, then that's basically describing the whole procedure. So it's very natural to say, well, let's base all the things with a subscript 1, which is like the stage 1 stuff, on the data that we have at the interim analysis, the overall survival data that's been collected so far. And then the things with the 2s in, we'll say that comes from the new additional data about each treatment comparison. Suppose at the interim analysis we decide to choose experimental treatment 1. And that's what we're going to go on with. So actually we lose interest in H02. We've dropped that treatment from consideration. So we don't even need to define Z22 because we're not needing it to test H02. But we do have to carry on testing the combination, the intersection hypothesis, which says that both H01 and H02 are true. Well, you can actually base that just on information about treatment 1 versus control. Right? That H01 is true under the intersection hypothesis, so this Z21 has the right null distribution. So that's quite legitimate. And if we'd selected experimental treatment 2 instead, then we get the mirror image, that we're not following hypothesis number 1 anymore, and we can base our intersection test on just more experimental treatment 2 data. So let's do that and make all of these Z standardized Logrank statistics. And at the first stage, actually the first interim analysis, testing the intersection hypothesis, we can just pool the subjects on treatment 1 and treatment 2, do a combined test of that group against control. A Logrank test would have a suitable distribution. So that's nice and easy. So just to reiterate that, at the interim analysis, we have a Z statistic for treatment 1 versus control, just a Logrank test of the comparison of those patient groups. For treatment 2, we've got a similar sort of comparison. And then for the intersection hypothesis, we've got the pooled treatment 1 and treatment 2 compared to control and a Logrank test there. If we select treatment 1 at the interim analysis, then in stage 2, we say, for the second half of the combination test for H01, we look at the additional data on experimental treatment 1 versus control. And that result about the Logrank statistic says that you get an independent increment. That's what we're relying on. And then to test the intersection hypothesis, we just say, well, we use the same statistic as our stage 2 contribution to that test. And if treatment 2 is selected instead, then everything just swaps around, 2 replacing 1. So I've called this is method 1. And if you remember the summary, this was the method that isn't going to work out. So what's going to go wrong? Well, there's nothing wrong at the first stage. The interim analysis, that's okay. But what about in the second stage? Think first the red bit. The extra data for testing the H01 about experimental treatment 1, well, that's got the sort of problem that Bauer and Posch raised. The way that we continue the study, and we might be tweaking the stage 2 a bit, making it bigger or smaller or more follow-up, longer accrual, who knows, could be influenced by the earlier information on the first cohort and their progression-free survival, and then that feeds into the follow-up of those patients. Now, you might say, well, that's okay because I laid down what I was going to do and I'm following the rules. And you might convince the regulators that that's the case, but they're a skeptical lot. They'll challenge you. That's quite a subtle problem. If we just move on to the blue one, I mean, forget whether you can convince the regulators that you're okay. Things go badly wrong here. And this is a much more obvious problem. If I'm setting my intersection hypothesis test statistic in stage 2 to be Z21, I'm automatically saying I'm going to test that with a data set where I've got these patients who did well in the first analysis on treatment 1 rather than control, like say in terms of progression-free survival. So you'd think they'll probably show better overall survival again. So we're going down this route because we're in a situation where we know this Z21, is going to be a bit better than average. Otherwise, we'd have chosen the other treatment. So this is clearly biased, and there's no way you can say you're doing something subtle to. Yeah.
Made the decision analysis only on the basis of the rather than
Sure.
the progression. Bias still
No.
persists?
No, so that's where the independent increments property of the Logrank statistic would actually apply. If the only thing you knew about was overall survival and the Logrank test, because the patients who've contributed to that, well, either they've died, in which case you're not following them anymore, or they've simply survived so far, and under your null hypothesis, they're going to carry on as normal. It's the fact that you've got extra information about them, which tells you more about how they're likely to fare in overall survival now because you've seen their progression or not. Or you've even, you could have other data, performance status, lack of adverse events. I don't know, that might be another issue. So this really is not a satisfactory method. And I should say my contribution to the work with Jenkins and Stone was to point this out. Although Bauer and Posch had pointed it out, I was just relaying that information. And so Martin Jenkins had a very neat idea to say, well, let's just slice things up in a different direction. So what we've been doing in method 1 was saying, we had this stage 1 cohort. They contribute information on overall survival during stage 1, and then they carry on with more follow-up, contributing information in stage 2. And we were saying, well, Z1 is what we know at the interim analysis. So that's just got this part. Z2 is what we learn after the interim analysis. So we've got the further follow-up of the same patients and the new patients. And we've put those together. And it's the fact that this stuff is somewhat predictable based on what we know at the interim analysis, and yet it's going into the stage 2 statistic that's the root of the problem. If you say instead, well, let's just define Z1 to be what we get from all the overall survival on the whole cohort admitted before the interim analysis, but actually including their later follow-up as well, then you get a nice separation. So Z1 is based on the first group of subjects. Z2 is based on the second group. And these will be conditionally independent, given your interim decision. And what this means is that the problems go away. So in this reorganized approach, we're used to calling it the stage 1 statistic. When you talk about combination tests, you have stage 1, stage 2. But it's not what you learn in stage 1, it's what you learn from the stage 1 cohort. And that's the key difference. You still have to be a bit careful and say, well, the total amount of follow-up for that first cohort mustn't be influenced by something that you learn at the interim analysis that might allow you to make some adjustments. So you've got to say at the beginning, I'm going to follow them up for a total of, 4 years, or until there are 150 failures. And we then get a test of H01, H02, and an intersection test. And then for stage 2, we just do the same thing with the second cohort of patients, and there'll only be one of these 2 treatments involved. So if it's treatment 1, we have a Logrank test for treatment 1 against control, and we use that Z also to do the second part of the combination test for the intersection hypothesis. Okay. So that's really all I have to say in describing this method. A little bit more talking around it. The paper that we wrote was describing a problem with an adaptation which was to do with, do we follow a full population or a subpopulation? I've couched this in terms of treatment selection. I think that's maybe a slightly more accessible problem. And there is actually a trial running with treatment selection very much along these lines using this approach. There is still a small print. We've got to be very precise, and in our paper we had some very strict referees, and I think that was a good thing, who made us spell out exactly this point, that you can't even say, this mattered more in the subpopulation problem. If you go to the subpopulation and you only recruit the subpopulation, it might take longer to get the patients in the second phase. You therefore might have, you might end up with more follow-up on the stage 1 cohort. You can't use the extra stuff. Right? There has to be one rule for what you'll do with those stage 1 patients. Okay. So that's a working method that can be used. There are a couple of references I'd like to mention that relate. There's a paper that came out later than ours, Earl and Schaefer, and it's actually doing very similar things in the same context. The point of presentation is that whereas we've talked about combination tests, they talk about using the conditional probability rejection principle, which in many ways is almost the same thing. I think the combination test is a bit simpler because one thing you have to go through with this approach is to say, well, if we carry on and do what we said we would do, we do a probability calculation, how likely is it we would end up rejecting the null hypothesis? And that means you've got to say, well, how many failures will there be? What will the information be, which is a random variable, which you don't know. And so you have to make something up. And I'm not sure how you make it up, but they seem to think you can. I think the combination test deals in a slightly different way with the same problem in a way that is much more straightforward. And another paper to mention, and the et al. Include Sue, I forget who else is in that list, there's a very similar, well, it's the same approach to the slicing of the data going on here. Is this something you're going to talk about, Sue? A little bit, so I won't say too much. The setting is not survival, but it's longitudinal. There's a short-term endpoint and a long-term endpoint. So exactly the same issue will arise. If you make an adaptive decision based on the short-term endpoint, then that's probably predictive of the long-term endpoint that you're going to be testing later. And Sue and her co-authors slice the data up in the same way. They talk about the first cohort and the second cohort rather than what we know at the interim analysis and what we learn later. So my conclusions are that you can put together the ingredients of an adaptive multi-arm trial for survival endpoints. And it's certainly desirable to do that. I mean, I haven't tried to assess the value of all this in this talk, but clearly it's possible to get benefits from the two-treatment versus control trial. And the machinery of a closed testing procedure and combination tests can be used, but some care is needed. And the independent increments property can fail. And actually, that wasn't necessarily the key thing. It was the way the intersection hypothesis was being tested, which was much more blatantly a problem. But if you define those in the right way, you can have a valid testing procedure. Okay.
Thank you very much for the very nice presentation. I'm Margarita Donica from Novartis. I just had a very quick question regarding the method 2, where we would only use the, let's say, the combined data from stage 1 and the follow-up of the first cohort. In practice, I'm wondering if we take a decision to drop a treatment, most likely in the clinical trial it's going to be difficult to continue giving this treatment to patients. So we will still influence somehow the outcome at the end of stage 2, because once we take the decision at interim, so let's say treatment 1 is no longer believed to actually be positive, so patients will go off treatment. And so we will influence the outcome at the end of the follow-up of these patients still, right? Wondering if that has any impact on the second method.
Yes, well, I think it has an impact on both methods. So my assumption here was that this whole cohort would be continued to be followed up. Would there be a treatment change? It somewhat depends on the nature of the treatment. If it's a chemotherapy that's given for several cycles and then that stops, then the treatment could be complete by the time this interim analysis decision comes out, and it's simply a matter of follow-up. Because it may be that having selected one of the treatments, you might then say, some of these patients want to switch altogether. Well, so the example, I guess the example I gave was rather fortunate. It's basically the same drug here, but it's been administered either rapidly or slowly. And it's already been administered. So I think if these were two different drugs, that would be more of an issue. But clearly that's a point. There is an assumption here that, yes, the treatment
In a way,
doesn't get to it.
Maybe it's even sometimes worse in oncology, because if patients progress, then often they go to another treatment. The progression itself triggers third-line therapy or whatever.
But that, I think that could be, if you take treatment as the broader strategy, then that could well be in the treatment definition, that after progression, then it's the physician's choice.
I was just wondering, you said you don't see an issue with the inability of the second design to stop at interim. I don't know.
Oh, sorry.
What would you recommend if you want to retain the possibility to stop at interim for efficacy?
Okay, so the second method can't do that because the Z1 only
Yes,
we learn about it at the same time as
yes,
Z2.
exactly. So you have to say from the start, I'm going to go right till the end. There is no chance for early filing.
Yes, I wonder. I think I mean it depends on the rates of progression and survival. It's quite likely that I think you just wouldn't expect to have that sort of evidence because there'd be so little in the way of overall survival data at the interim analysis that you'd ignore that option. So what would you do if you... I'm not sure. I wonder if you could take the Z1 in two parts. Could you'd have you could look at overall survival Logrank test, which will be the first part of Z1 at the interim analysis. I'm just trying to think on my feet. I think if you had enough data and you had enough extreme results, you could make a stopping boundary based on that. That would be legitimate. So you'd then have a sort of Z1 part A and a Z1 part B and a Z2. So I think that's doable, but certainly the situation that the trial I'm aware of, I'm sure that hasn't come up because they're not expecting much in the way of overall survival data at the interim point. I mean, there may also be issues of total numbers of patients that you want to have on the treatment and safety information and so on as well. Yeah.
How easy is it to say how long you're going to follow the before you even see any data? Follow this first cohort for this period of
Well, so the problem is you might say I'm going to follow them for 3 years, thinking that will be long enough to get so many events, but actually you don't get that many events. It could be the baseline survival rate is better than you thought. But you can define that amount of follow-up in terms of events if you prefer, or both. So you could say, well, it'll be 3 years as long as that delivers at least N events, and if not, we'll keep going till we get N events with a maximum of 5 years, or as long as it's a definite rule. That's the main provision that's needed.
cohorts or stage cohorts. How do you
Well, for survival data, everybody overruns. I mean, there's always more data to see. So I don't think this is necessarily any different from simpler survival studies where you can have a stopping rule and then you'll, assuming that the treatment isn't becoming irrelevant due to unblinding and so on, you get more follow-up that could still be informative. So I don't really see anything special. Maybe with Sue's example with longitudinal data, where there's a definite time at which you look and you can have, because of the delayed response, possibly an interim analysis, and then you get, depends how many analyses you have. You might want to stop and then you get more overrunning data. Well, I'm very interested in that problem in its own right in the group sequential setting. I haven't thought about it with multiple arms. I guess the approach would be to try and take the pieces apart and think about it one hypothesis at a time, which is the nice thing about the closed testing procedure approach, that you've got this multiple testing problem, but you break it down. No, that's a food for thought, that one.
People to say, well, I'm only going to use the Logrank overall survival.
Yeah.
I'm not going to use the progression-free survival. Could say, well, I'll put N patients on at the first. At some point, I'll stop.
Yeah.
And I'll use all the data.
Yeah.
To do the final analysis. Here you'll have to say, well, I've pre-specified how many events I'm going to use from that.
Yes, unfortunately, for the type 1 error protection.
Giving up that flexibility.
Yeah.
Is there a trade-off that maybe you can say? I mean, it depends on how closely progression-free survival is correlated with overall survival, right? You could give up on say I won't look at. I mean, forget about regulatory authorities. You could say I won't look at progression-free survival. I'll only look at overall survival,
Sure.
and I'll get more flexibility that way.
Yeah.
Have you considered what's the trade-off between getting more flexibility, all the data at the end versus extra?
I think the serious problem is having useful information at the interim analysis to make a treatment selection. Right. So, if overall survival is quite good, then basically all the data you have at the interim analysis, or 90% of it, is to do with progression-free survival. So it wouldn't be an option to say, well, let's just use this tiny amount of overall survival data to make a treatment choice. But I mean, if you take a different indication or different, it's also the accrual rate, comes into the equation. You could change that. I think what you lose in this particular case, I don't think you are losing much here in that the total time until the end of the study, it might vary a little bit with recruitment, but it's broadly fixed. It's rather different in a case when you have not a change of treatment, but a change of population. Because there you would definitely, if you go to the subpopulation, then accruing the right number in this stage from a smaller class is going to take longer, so you'll have more follow-up, and you would have more follow-up here that you then have to discard. So you could try and look at the trade-off, and well, if you simply can work with overall survival, then everything's a lot easier, definitely. But I think it's the interim decision is pretty noisy anyway, so you probably and the other point you make is very important. That it has to be a good surrogate, which I think that's not the regulators don't take you to task about that, but you might worry yourself if you're.
Chris, thanks for the excellent presentation. I have a question to slide number 17. Because you have said you simply split it up in the first and in the second cohort. And if you drop a treatment at the interim analysis, you still haven't observed the pooled Zed statistic of the first cohort. So this means if you have a long-term treatment, what do you do with the patients which are in the drop treatment arms to get the test for the intersection? It would mean you still would have to treat them. Otherwise, you won't get your test statistics.
No,
so I think there's still an open point.
Right. So the assumption here is that you carry on treating them. I think this was one of the first questions was very similar. And in some cases that would make sense, and I think in other cases that might not be practical. So that needs another solution. if that's really the case. Yeah.
Targeted therapies today, you don't really have a duration anymore. You treat them until progression or lost to follow-up. So you no longer, you really would have to treat them. And if we drop one, patients will no longer be willing to continue, or even the investigators will no longer be willing to give that treatment, which we discard at interim. Then we would absolutely influence the second stage.
So, I mean, you could, I suppose you could simply truncate everything on the first treatment to shortly after the interim analysis on the grounds that, that's as far as the follow-up will go. What you're losing then is the further follow-up on those patients on the treatment that you keep going. They are providing more data, but I suppose, and that's the kind of data that it's very hard to know what to make of because it's automatically biased because you chose that treatment because those people were doing well. So maybe there isn't, there's no way round. No obvious way around how to keep using that data.